a question that may seem kind of silly, but almost every little kid asks at some point. They say, when I see green, maybe it's not the same green as you see. Maybe when I'm seeing green, you see red. And so little kids are thinking about how our brain works. We started off this purely because of curiosity. Color blindness is the most common, we could call it genetic disorder in human beings. It's kind of amazing because not only is it the most common one, but it's more common than all other single gene disorders in man. 1 out of every 12 men have a red-green color vision defect, but they're kind of lumped all together.
Hold your eye open.
It turns out that some people that have a red-green color vision deficiency actually see red and green quite well. They just see things differently than the rest of the world. They can tell the difference between colors. And then there are people that are missing a red or green photopigment gene, that are missing a dimension of color that would make a huge difference in their lives. At least that's what they tell me. If they, had that photopigment that other people have. They're really missing the ability to distinguish a lot of different colors from gray. And the only way to make them be able to kind of appreciate the colors that a normal trichromat would be to give them the extra capacity. Well, this is where we do all the DNA processing.
I'm pretty sure that you don't have the green gene because I actually specifically remember your sequencing, but I remember that when I tried to amplify the green gene from your sample, I couldn't. When you came, actually it was in the very beginning of the study that I was running. So I was still, very new to this, and I hadn't seen every particular situation in every particular case, and because my project was focused on people with normal color vision, I wasn't actually used to running all the people who are colorblind. So, when something weird popped up, I was like, I cannot get this guy's green gene to amplify. What's wrong? What am I doing wrong? It's like, well, duh, because he doesn't have one, So I actually specifically remember because you were one of the first colorblind people that I had personally done, the whole sequencing and everything on, and so it made perfect sense. It's always nice when something doesn't work for, because it shouldn't. But we can verify it. We can actually go back and look at see what you have. Oh, actually, you're not in the group. You're in the. Yeah, see, I have your folder, and right here I wrote that I got no green product twice. I tried it twice because I thought I did something wrong, and it confirmed the mass array result that you are L, so that you don't have a green gene. This is kind of what we do. These sites that are highlighted, these are the variable sites. And so these are the sites that determine the sensitivity of the pigment. And these are the sites that vary among people. So for each subject, we go through and we kind of figure out what people have at these particular sites. So yeah, so this is you. So there you are.
There's a difference between being different and not having an ability. And I think that that's really important. It's something that I'd like to kind of help communicate to the world. People have heard that it's so much more common in men, but it affects 1 out of every 12 men and affects about 1 out of every 230 women. Probably everyone has known a woman who's colorblind in their life, but that colorblind woman didn't necessarily say, hey, I've got a problem with my color vision. We developed a method where we could actually record electrical potentials generated by the retina where the cones are.
Gently lay the really, really thin nylon fiber right across your eye. It shouldn't be painful at all.
Is it okay if I blink?
Yeah,
Okay.
it's totally fine. And I'm going to pull down on your eyelid. Just hold your eye really, really wide open. All right, how's that feel?
A little weird.
Yeah, but not painful or anything.
No, not at all.
We can put a little fine electrode just on the sclera of the eye. People blink over it, it's no problem. And then we shine lights of different colors in the eye. And so we can shine red light into the eye, and then we can look to see if the eye will respond with an electrical potential. And this can be done in such a way that you can actually get a map of where the potential is coming from. We said, gee whiz, now we have lots of things that are the perfect combination to try gene therapy. One, we have a closed vessel. We could put a solution containing a vector that would have a photopigment gene, and we could put it in the eye, and it would stay in there, and it has to go then to the photoreceptors. And we said we could pick out a primate model that has red-green color blindness just like humans, and then we used a human red photopigment gene and put it into a viral vector, and we made an injection into the monkey eye, and the actual DNA was taken from a virus, and we used the virus's capability to attack a cell and inject DNA into it. But the DNA it's now injecting is our therapeutic gene. We made monkeys that had started off just having blue cones and green cones, and then we turned a subset of those green ones into red ones. So now they had red, green, and blue cones. And now, what we're interested in is how long does the therapy last? Still, those animals get up every morning and do their color vision test, and it's been now more than 4 years since they were treated. Having successfully cured color blindness in monkeys means that it's possible to cure color blindness in humans. I am pretty sure that if we did exactly the procedure that we performed in the monkeys in a person, it would cure their color blindness. What our challenge is to be able to develop the procedure so that we could give people the benefit, but be sure that the risk is effectively zero. And we believe that this will be possible.
We have a couple goals, and one of the biggest goals is that we would like to develop a virus that has the gene therapy vector. So take the virus and then take a gene that we would like to express. And we would like to be able to inject that virus into the intravitreal space, so inside of the globe of the eye. And we would like it to diffuse freely and to pass all the way through from the ganglion cells all the way back to the photoreceptors, because the retina is built backwards. The actual light sensitivity is in the way back part of our eye, and then the retina has different layers that build on top of that. So we would like to be able to put this virus into the intravitreal space and have it diffuse to the cone photoreceptors, into the rod photoreceptors.
We looked at the DNA of women, and some women actually have 4 different kinds of cone photoreceptors. So the idea that people have the retinal basis for tetrachromacy is a proven fact. But what their visual experience is an open question. You know, it's possible. That's really what's going on, is that all of these women that have the extra kind of cone in their eye, if you trained them, that they could learn to see this extra dimension of color vision, and they just haven't had the experience.
This is one of the fun things that I get to do in the Neitz laboratory, is I get to use my electrical engineering undergraduate degree to handle and to actually solve some of these problems. We definitely are always needing new technology that doesn't exist yet. A project that we're working on is, of course, a projector that can produce 4 different primaries instead of 3. This is still in its development phase, but ultimately we're going to illuminate this mirror with 2 light sources, and we're able to adjust the lights such that this new photopigment that we have not introduced yet, we're going to do one before we do the injection, and we're going to be able to see that there's no response of the retina at all in order to those particular lights that we used. But if the tetrachromatic injection works and their retina gets a new sensitivity, we'll actually be able to see that suddenly there was no response before to the particular lights that we used. But now, because the retina has a new sensitivity in it, suddenly the same lights will elicit a response. And because it is a multifocal ERG, we'll be able to actually see exactly where the new sensitivity essentially landed.
If we can take a monkey that has 2 photopigments and give them an extra dimension of color vision, from dichromacy to trichromacy, what about adding a 4th photopigment and seeing if it's even possible to expand the visual experience?
This person who is a tetrachromat, they wouldn't have the same activation of our red and our green and our blue cones. They would have another activation in there that would be slightly different, and theoretically they could be taught maybe to see something that they've always, maybe it's always happened where some normal person says that's white, and they've always said okay and accepted it, but maybe it hasn't been white to them.
What is our capacity in order to absorb the new kinds of information that come from a new input? And I think this is a useful thing to do. And at least we could begin to think about the possibility that maybe we do want to have an extra dimension of color vision. I know I kind of do.